Hey guys, so today we're going to talk about the cell theory and some of the characteristics that all different cell types have. The first thing that we need to talk about is what is a cell? Well, a cell is the smallest unit of all living things, meaning that anything below a cell is not technically considered living, so things like atoms, molecules, or even organelles. Um, and we have come to understand what's known as the cell theory based on the works of several scientists. And some of these scientists are listed on this slide here. So pretty much we've developed this theory over about a 100-year time period, and it was developed about 200 years ago. So this idea has been around for a very long time, and it was composed of three main parts. The first saying that cells are the fundamental units of all life, meaning that all living organisms are going to be composed of cells. So the first and second part of the cell theory kind of go together. And then that last part of the cell theory states that all cells are going to come from pre-existing cells, meaning that they cannot just spontaneously appear. All cells are self-reproducing, meaning that they can undergo mitosis or a self-reproducing cycle to produce more cells. So one thing that you should be asking yourself now is, why are we composed of so many small cells as opposed to really large cells? Well, that has to do with a surface area to volume ratio, and we're going to break that apart. The surface area of a cell determines how much of a substance we can eliminate or allow into a cell. And the volume of a cell determines how much metabolic activity per unit of time that cell has to expend. So what this means, if we look at it in the form of a diagram, is that a larger cell is going to have a very large volume, which means it's going to have to perform many processes and expend a lot of energy to accomplish everything it needs to accomplish. It also has a pretty high surface area, which means that it cannot quickly eliminate waste out of the cell, or it cannot quickly allow materials into the cell if it needs those. If you compare that to a smaller cube, which would be more ideal for a cell, you should notice that it has a pretty high surface area to volume ratio when you compare it to the larger cell. So a high surface area to volume ratio means that a cell can quickly eliminate waste and quickly bring materials into the cell. It also means that they can expend less energy and accomplish everything that they need to in order to perform its daily functions. So the key point here is that a high surface area to volume ratio is important and is what makes cells stay very limited in size. On the right here, I just gave you a diagram to show you some of the different sizes of materials and what type of microscope you can use to observe these materials or organisms. So how do we study cells? Well, you might be already familiar with the light microscope, which is pretty much what we're going to use all year long, or a compound light microscope. And these types of microscopes use a combination of glass lenses and visible light, and it's that visible light that allows us to detect certain structures within cells. We can see organisms as small as 0.2 micrometers in diameter, which is about 1,000 times smaller than what your naked eye could see. So these are pretty efficient in just a general cell structure or visualizing different cells. Contrast to that is an electron microscope. And these are microscopes that are going to use a combination of magnets and electron beams to observe either the internal structure of an organism or the overall 3D shape. And if you're viewing the internal structure, you're going to use what's called a transmission electron microscope, known as TEM. This allows you to shoot electrons through an organism and to view that internal structure. The scanning electron microscope, or SEM, allows you to see the overall surface or 3D structure of an organism. So along the bottom of this slide, I've just given you three examples of the same organism viewed under different microscopes. Here's a euglena under a light microscope, so we can see its overall general shape. This is viewing through a euglena, so that's a transmission electron microscope image. And the last one on the right is a scanning electron image of a euglena, meaning that we can now see an overall 3D shape on that diagram. Here are just a couple of pictures to show you what these microscopes look like. On the left is a compound light microscope. Make sure that you can identify all of those parts pretty quickly. You should be able to remember that from biology, and if not, we're going to do a little bit of practice, but you should have a basic idea of some of these different names. On the right is what a typical electron microscope would look like. These cost billions of dollars, and they can be much larger than what you see in the image. A lot of universities have an entire floor dedicated to electron microscopes. You can only handle them with certain equipment because you are dealing with electrons and you don't really want to shoot electrons through anything living. So those microscopes are only going to be used for preserved or dead organisms. And the last thing I want to talk about is, what do all cells have in common? So you might recall that there are two types of cells, prokaryotes and eukaryotes. Prokaryotes are things like bacteria, and eukaryotes are pretty much everything else. Well, regardless of the cell type that we're talking about, these cells are going to have some similar characteristics. They're all going to have a cell membrane, which is composed of a phospholipid bilayer. We're going to talk about that a little bit more in detail, but just make sure that you can describe that a cell membrane is composed of that. And also, these cell membranes are selectively permeable. And what that means is that they can choose what enters and exits the cell, which is really important. All cells are going to have ribosomes because this is the site of protein synthesis, meaning where all the proteins are going to be made. They can be free-floating in prokaryotes in the cytoplasm. They can also be free-floating in eukaryotes in the cytoplasm, but you're going to find more in what's called a endoplasmic reticulum. And in a couple of videos, we'll talk about cell organelles. They all have a cytoplasm, which is that jelly-like substance in the middle holding everything together. And they all have DNA, which is the hereditary material that's needed for all living things.